Good day, everyone. I hope you're having a great day wherever you are. Today, just join me for the topic in licenses. The first chapter called Nucleic acids, created by me, right? And then we're going to learn about section 1.2, which is DNA replication and its significance. Remember, we had 1.1, and then if you didn't watch 1.1, go back to the previous video so that you can watch it. DNA replication, right? And then what happens is that what you need to know first is that what is DNA replication, right? And then where does it take place? So basically, DNA replication takes place during what? During interphase. It takes place during interphase, which is before mitosis and meiosis begin. So it means that interphase is a phase that begins before mitosis and meiosis begin, right? So what I'm trying to say here is that interphase is an old phase. It comes before mitosis and meiosis, right? And then what you need to know is that you need to know that the reason being is that we have something called IPMET. We have something called IPMET. I don't know if you heard about it. IPMET. IPMET. This is protein that is there. That is A, and then that is T. That is T, guys. That is T. Let's just pretend that is T. So what happens is that interface comes before IPMET. What I mean by this abbreviation is that I is for what? I is for interface, which comes first, and then prophase. What happens is that prophase. This is the prophase. P stands for prophase. M stands for metaphase. A stands for anaphase. T stands for telophase. So the first one is interphase. It's the first phase, right? And then DNA replication is what? DNA replication is a process in which DNA makes an exact copy of itself. So like DNA makes an exact copy of itself. It's an exact copy, not a different copy of itself, but an exact, the same strands, the same nitrogenous bases, the same everything, exact copy of itself. And then what happens during DNA replication? The first stage is that DNA double helix unwinds. So we'll be having DNA double helix, which unwinds in which way, right? In which way is that, like, we're having something like this, guys. Let's just pretend this is a DNA molecule, right? Let's just pretend this is a DNA molecule, right? And then what happens is that, like, this is a DNA molecule. What happens is that it unwinds in such a way that then then nitrogenous bases, they split, right? And then what happens? They split. It unwinds, and then so and so on, guys. And then the nitrogenous bases in between, they unzip or break the weak hydrogen bonds. They do what? They unzip or they break, right? Because they are weak, right? Weak hydrogen bonds. Weak, meaning that they can be easily broken, right? And then you'll check more on that, guys. And then we have what? With the significance of DNA replication. The significance of DNA replication. The first one is that it doubles the genetic materials, so it can be shared between the resulting daughter cells during cell division, right? And then the other one is that it results in the formation of identical cells during mitosis. So, like, during mitosis, it results in what? In the formation of what? Identical cells. That's what you need to know. And as always, guys, thank you for watching. And then up next is section 1.3. You'll enjoy. Thank you.